audience, welcome back to Naeem Bukhari YouTube channel. So today we are going to discuss variable gene expression. This is lecture number 9 from human genetics subject. So we are going to discuss variable gene expression, means how genes are expressing in a varying fashion. As you people know, all the humans have same genome, but the expression of genome is different in all the humans. That's why humans exhibit different sort of their characters, and their bodily structure are differently made because their genome expresses in their body or in their cells in a different way. So we can say expression of genes in a set or variety of ways actually decides the bodily patterns, okay, in the variation within an individual as well. Okay, in this lecture we are going to discuss the contents. These are major contents which we will discuss. The contents includes like introduction, definition of the topic and further objectives, identification method, and example cells and conclusions. Okay, these things will be discussed in variable gene expression. So first we are going to move for introduction. In genetic, expressivity is the degree to which a phenotype is expressed by an individual having a particular genotype. अब हम पहले इस बात को समझेंगे कि फिनोटिपिक इफेक्ट्स जो होते हैं या एक्सप्रेसिविटी क्या होती है. तो एक्सप्रेसिविटी से मुराद ये होती है कि इफ़ देयर इज सेट ऑफ अ जीनोटाइप इन जीनोम ऑफ एन इंडिविजुअल सेल, then we can say that genotype will be fully uncoded into a phenotypic effects, okay, and then that phenotypic effects we can visibly see or we can observe, okay, and those phenotypic effects appear in the form of proteins because once gene sequences are uncoded, those uncoded gene sequences will be translated into the proteins, okay, and the proteins can visually be seen in cells, okay, and once the proteins will be produced in the cell, so cell exhibit different morphology, their structural components, and we can say and their overall function as well. Once the diversity or variety of protein exists in cells, cell will also have different sort of functions. So now further expressivity is related to the intensity of a given phenotype. If we want to discuss means how much a gene is expressed, that is directly related to the phenotypic effects. Means if a gene is fully expressed, means its phenotypic effects are also intense or are also observable with greater intensity. So we can say expressivity of the gene is actually directly linked with the phenotypic effect appearance. Range of signs and symptoms that can occur in different people with the same genetic condition, okay. This is a different and a major aim of this topic. Means all the human have a same genomic makeup, but those genes express in different fashions, which results in sign and symptoms to whom people are different in. Okay, all the peoples are having different bodily morphology because the same genome in all the individual is expressed in a different fashion. That's why human beings are representing a diverse, we can say, morphological appearances and the cellular function or functional appearances as well, okay, or functional diversity. Here we can say X variable in mathematics. So this we are just supposing, I'm just quoting X variable in mathematics as an example for you guys, just to understand the variables in gene function or variability or diversity in gene functions. Okay, we already expressed, we disclosed that variable gene expression will be expressing themselves in a variable fashion. Like, what is the reason why genes are expressing themselves in a varying fashion? Means if an individual have same genomic makeup and other individual with same genomic makeup, but they are they are showing different phenotypic effects with the same genomic makeup. What is the reason? So all the reasons are not actually, scientists are unable to disclose all the reasons why this happens, actually why genes produce different proteins or different products, or why genes are differently expressed in different individuals. Scientists cannot disclose this in proper. Only some of the factors were proposed, so we will discuss those factors which are responsible for variable expression of the gene in the next slides. So that's why we expressed as in mathematics, once we don't know any parameter, we propose that parameter with the X. Same in this fashion in genetics, we propose those genes whose functions are whose a variable and their functions are restored from time to time, or they are expressed in different fashions, then we express the reason is actually x, means the reason is varying from individual to individual, right? Similarly, antibodies. In antibodies, there are variable regions and constant regions. Variable regions can bind with the number or variety of antigens. Similarly, in human genes, there is a great varying, we can say constant, means genes will be expressing themselves in a varying manner, okay? So genes varying manner also suggests us, like if genes will be expressed in a different way, then a different sort of proteins will be produced. This is the ultimate story, means how genes are expressing in a variable fashion. Further, we also discuss what are the reasons, what are the main objectives of gene expression in a varying fashion as well. Okay, now moving for the objectives, means if genes are expressing in a varying fashion, then what will be the most important goal of single cell RNA sequences is to identify highly variable genes across a population of cell to account for the discrete nature of single cell gene expression and uniqueness of the sequences. Okay, अब हम लोगों का बेसिकली गोल क्या होता है? वंस वी आर गोइंग टू असेस वेरिएबल जीन एक्सप्रेशन, सो हाउ वी कैन असेस दैट वन. तो हमें क्या चाहिए कि जैसे हर जीन है, हर सेल है, तो हर सेल के अंदर उसका जीनोमिक मेकअप मौजूद है. दैट जीनोमिक मेकअप इज एक्चुअली एक्सप्रेसिंग दोज जीन्स इन अ डिफरेंट वे. जब जीन्स डिफरेंट वे में एक्सप्रेस होते हैं तो उनके प्रोडक्ट्स भी डिफरेंट बनते हैं. अब हमने अगर जीन्स की जो एक्सप्रेशन को अगर स्टडी करना है तो जाहिर बात है हर एक सेल को हमें स्टडी करना पड़ेगा, बिकॉज हर सेल में जीन्स जो है वो डिफरेंट तरह से एक्सप्रेस हो रहे हैं. और डिफरेंट इंडिविजुअल में फिर बहुत सारे सेल्स मौजूद हैं, तो डिफरेंट इंडिविजुअल्स की जो डिफरेंट सेल्स हैं उनमें भी जीन जो है वो डिफरेंट मैनर में जाके एक्सप्रेस हो रहे हैं. इस वजह से एक बहुत बड़ी ग्रेट डाइवर्सिटी एग्जिस्ट कर रही है जीन्स के जो एक्सप्रेशन पैटर्न्स हैं. दैट्स वाई अब हमें क्या चाहिए कि हमने क्या करना है कि सिंगल सेल आरएनए सीक्वेंसिंग करनी है हर एक सेल की, तब जाके हमें पता चलेगा कि हाउ जीन्स आर एक्सप्रेसिंग इन अ डिफरेंट और वेरिंग मैनर. अब हम ये कैसे, वाई वी डोंट गो फॉर सिंगल सेल डीएनए सीक्वेंसिंग, वाई वी आर प्रेफरिंग सिंगल सेल आरएनए सीक्वेंसिंग? उसकी वजह ये होती है कि जब किसी भी न्यूक्लियर मटेरियल से जो आरएनए बनता है, बेसिकली एमआरएनए बनता है, जब वो एमआरएनए न्यूक्लियस में बन के साइटोप्लाज्म की तरफ मूव करता है, तो साइटोप्लाज्म की तरफ मूव करता है तो वो आरएनए एक्चुअली कुछ प्रोटीन बनाता है और साइटोप्लाज्म में वो प्रोटीन्स बनती हैं फर्दर, लेकिन उनके लिए जो इन्फॉर्मेशन है वो एमआरएनए के पास होती है. अब अगर सेल की प्रोटीन्स की बात करें तो सेल की प्रोटीन एक्चुअली एमआरएनए मैसेंजर आरएनए के पास होंगी, जो इंस्ट्रक्शंस हैं, जो सीक्वेंसेस मौजूद हैं. तो इसी वजह से हमने क्या करना है कि उस सेल के अंदर जो न्यूली प्रोड्यूस्ड एमआरएनए है विच कंटेन्स जेनेटिक इंफॉर्मेशन, विच कंटेन्स जीन्स to whom the proteins will be produced. So that's why we need to study that cell single cell RNA sequences, mRNA sequences actually. Then we can find the exact expression pattern of the subjected gene. Okay. Further, next term we have, I just added this term for your understanding, penetrance. अब penetrance से क्या मुराद है? How a phenotypic effects exactly matches with the genotypic. Suppose if we say there are 10 sequences of genotypes, means if there is a gene and there are 10 base pairs present in a gene, suppose, okay, so it's mean if there another cell contains 5 pairs of bases, so the gene with 10 base pairs will be having more stronger phenotypic effects rather than a gene with 5 base pairs. And we can say if a gene with 10 base pair is wholly expressed, is completely expressed into its phenotypic effects, means genotypic sequences are translated into phenotypic sequences in a complete fashion, this is known as penetrance. Okay, so it's also known as quantitative measurement of the genotypic effects into the phenotypic effects. This means penetrance. हम कहें कि एक जीन है जब वो completely express होगा तो उसकी जितनी भी bases हैं वो express होंगी तो वो phenotypic effects उसकी proteins वो बना लेगा. लेकिन एक जीन है वो क्या होता है कि वो उसके जो जितने sequences हैं उनमें जितनी information मौजूद है वो अगर express नहीं होता, अगर जीन express नहीं हो रहा तो इसका मतलब है there is a mutation and there is no completely genotypic sequences are expressed into phenotypic sequences, and this could lead towards a disease. Okay, where in variable gene expression what actually happens in variable gene expression, there are qualitative, we can say, effects. qualitative effects are observed in journal, because genes are expressing themselves in a varying fashion. Okay, that's why we don't go for the quantitative measurement of the visible effects, so we will be going for the qualitative aspects in case of variable gene expression. So these are some of the objectives we usually achieve while we are going to study variable gene expression in a cell. Okay, if you have any question regarding this one, post your question. I will try to answer your question. Stay blessed. Allah Hafiz.